Okay, so after we've got our pyruvate, we then need to add it to the Krebs cycle. And the whole, well, first of all it goes through the link reaction, and then we have the beautiful Krebs cycle. Krebs. And the whole point of all of respiration is, well, the whole point of all of this up to oxidative phosphorylation is to get as much reduced NAD as possible. Because that's how we can make our ATP during oxidative phosphorylation. Okay, so if I just go through this. So our pyruvate, we've got 2 of these per glucose molecule, and we're going reducing NAD again, and we're decarboxylating, so that's pyruvate decarboxylase, the enzyme. And this is the link reaction. It requires active transport to get pyruvate into the mitochondria, because it's got to get through these membranes. Okay. And then coenzyme A, or binds with the resulting acetate to form acetyl coenzyme A, and it's that acetyl coenzyme A which enables the acetate to bind to oxaloacetate, recycling CO coenzyme A, so that can just go back over there and keep on going. Okay, so we form our citrate, which is six carbons. So I'll just draw it 1, 2, 3, 4, 5, 6. And it's decarboxylated. So we're just going to do it really simply and remove one of these. So you have your 5 carbon. And at the same time as decarboxylating, you get the reduction of NAD, or hydrogenation. So it's the dehydrogenation of citrate and the hydrogenation of NAD, forming reduced NAD. Same thing happens again. Yep, get rid of one of these. And we've got our 4 carbons. And then, as part of A-level syllabus, it just doesn't really go into a lot of detail. So again, you have your NAD to NADH, which is dehydrogenation of this guy. Okay, so then you have substrate-level phosphorylation, which basically means that a phosphate from somewhere other than ATP is being used to make ATP, and it's likely to be that some sort of reaction that's going on with the formation of this releases an ATP molecule, because there's likely to have been phosphate groups added at some point, but you don't need to know any of that, so that doesn't matter. So that forms another 4C compound, but it means we get a nice bit of ATP, yay! And then we have FAD, which is like NAD, but slightly different, and that also takes electrons and hydrogens and becomes reduced FAD. And then we just need another reduction of NAD to form oxaloacetate. And that's the Krebs cycle. And just remember that the whole point is this guy, and then we've got more of them over here, haven't we? Yeah. Where's he gone? There he is. So at the end of the Krebs cycle, so this is per pyruvate. If you wanted to do it per glucose molecule, it'd be ×2 for all of these. So you have 3 NADH, 1, 2, 3, and you should have 1 FADH over here, and you get 2 CO2s and 1 ATP, like it says there. Okay, and then we go on to wonderful side of oxidative phosphorylation.